Welcome to the Institute of Medical Sciences, popularly referred to as the IMS. The IMS is part of the Forester Hill Health Campus at the university. It is one of the largest clinical complexes in Europe. As well as the IMS, it includes a state-of-the-art medical school, a large teaching hospital, the Institute of Medical Sciences, the Institute of Applied Health Sciences, the Institute of Dentistry, and of course, the internationally renowned Rawat Institutes. The IMS is the largest research institute at the university. Here, you find a thriving community of researchers and support staff who work on cutting-edge biomedical subjects aimed at understanding how the human body functions. My name is Dr. Peter Ihianusime Imoisi, and I'm a research fellow here at the Institute of Medical Sciences. And my research is focused on neurodegenerative diseases. To be more specific, Alzheimer's disease, which is the most common form of dementia. I started out my career at the level of PhD here at the Institute of Medical Sciences. During my time as a PhD student, I was able to establish the correlation between the derivative of vitamin A and its correlation between depression, suicidal ideation, and psychosis. Furthermore, during my PhD, I was also able to establish the link between a specific region of the brain that may be responsible for vitamin A balance in the body. Having said that, today I'm going to be your tour guide for the IMS tour, where you will have the opportunity to have a quick back scene of some of our core facilities and laboratory. And as well, you have the opportunity to meet some of our leading researchers who work on cutting-edge biomedical subjects. Do follow me, please. I'm here at the Atrium of the IMS. We have over 200 people who work here at the IMS, and we also host undergraduate and postgraduate students as well. I'm here at the Atrium, which is the main entry point of the IMS. Usually, this is a busy area where you have postgraduate students meet up to interact with friends and classmates. At the same time, we use this area for social and public engagement events. But due to COVID-19 pandemic, we are unable to have a group of persons into the IMS building today. And at the same time, we've had to set up strict regulations and measures to ensure that our staffs and research students are kept safe at all times. However, it would have been nice to have you here with us today for the European Researcher Nights. But for now, let's head into the IMS to meet one of our early career researcher, Dr. Ruta, who is also a research fellow here at the Institute of Medical Sciences.
Hi, I'm Dr. Ruta Dekirite, a research fellow here at the Institute of Medical Sciences at the University of Aberdeen. I work in diabetes research lab led by Professor Mirela Delibegovic, and my research is focused on finding new treatments for type 2 diabetes, obesity, and cardiovascular diseases. I started my journey on becoming a scientist around 10 years ago when I first came to do my bachelor's degree in neuroscience with psychology at the University of Aberdeen. I really enjoyed the quality teaching and research experiences provided to undergraduate students, and I then decided to continue on undertaking a PhD project where I was investigating the links between type 2 diabetes and Alzheimer's disease. I really enjoyed the project, and then I decided to stay here as a research fellow. I think the most rewarding aspect of my job is being able to discover something new and unknown. Every day is very unique and provides a number of learning opportunities. And I think it's just a very exciting feeling that you're running an experiment and you don't know what the outcome will be, but you are just, waiting for that eureka moment when you actually find something new.
Now, within the IMS, we have a wide range of research technologies which are accessible to researchers within the University of Aberdeen and where appropriate external bodies. These facilities include the Ian Fraser cytometry, which hold cutting-edge equipment to assist in the characterization and measurement of cells. We have the microscopy and histology, where we are able to look at samples such as cells, tissues, organs under the microscope. We also have as well the qPCR core facility, where we are able to examine the expression of genes in a wide range of samples. We are here at the Ian Fraser cytometry. Here you have researchers, support staff who are instrument technicians that provide support for researchers, training to ensure that these equipment are used safely and correctly. At the same time, they also provide support to external bodies who are also keen to carry out research within these facilities. What that simply implies is the fact that we do have access to these facilities as researchers, and at the same time, we are open to external body where we set up link with industrial bodies to also as well use these facilities for their own research and result analysis. The IMS is also a teaching location for our undergraduate students and postgraduate students in medical research sciences. Our students do have access to a wide range of these core facilities here at the Institute of Medical Sciences. This is why our medical courses are ranked the best or within the top 5 in the UK for student satisfaction. For now, I'm going to introduce you to one of our final year PhD students who currently works here at the Institute of Medical Sciences. Meet Charmaine.
My name is Charmaine. I am a final year PhD student at the IMS. I am currently working under the Rido and Platt lab, and I am collaborating as well with Atrium Pharmaceuticals in Finland. Currently, I am working on preclinical research in dementia. I am particularly interested in the reproducibility as well as the replicability of preclinical research. We are actually hoping that we will cultivate a culture that will encourage complete reporting as well as transparency in research, so that we could improve the quality of data that we have at the moment in scientific research, and as well as to pressure journal editors, researchers and peer reviewers to enforce methods of refinement, and hopefully that way our credibility as well as ethical justification in our preclinical research will not be undermined. I was studying anatomy and physiology for 4 years and graduated with a BSc with honors before I was recommended to come here and work for dementia studies, since I was particularly interested in neuroanatomy.
As earlier said, human health is the focus of research here at the IMS, beginning with cells and the molecules that make it up. The origin of almost all diseases lies at this core level. due to defects in genes, the protein they encode, and other micromolecular bases of cells. From this fundamental standpoint, clinical researchers are able to advance the cure. Activities within the IMS are focused on 4 main research programs. One is infection and immunity, where we try to understand how the body interacts to infection and the development of immunity. A classic example is the current COVID-19 disease. Secondly is molecular medicine, where we use and study physical, chemical, biological, and medical techniques to understand molecular structure and mechanism, identify molecular and genetic errors associated with diseases, and using molecular intervention to solve such problems. Third is system physiology, where we combine experimental and computational advanced studies in trying to understand how the human body functions. Lastly is translational neuroscience, where we apply basic or cutting-edge neuroscience technology in developing new therapies to address diseases associated with the central nervous system. And a classic example of such disease is Alzheimer's disease. Another researcher, Dr. Katrina Cunningham, is involved in translational neuroscience research here at the Institute of Medical Sciences with the Aberdeen Spinal Injury Research Team. Dr. Katrina is going to tell you more about our research.
I'm Katrina Cunningham. I'm a research fellow in the Aberdeen Spinal Injury Research Team in the Institute of Medical Sciences. Within my team, I'm postdoc, so I'm responsible for conducting experiments. I also help manage the lab on a day-to-day basis. I also help supervise and train the postgraduate students within our team. So my overall research aim is to develop novel combination therapies for spinal cord injury repair. My main project is focusing on a combination therapy that involves stem cells. So we use mesenchymal stem cells. They're a type of adult stem cell that are found in bone marrow and fat tissue. And instead of using the cells themselves, we're instead using secretome, which is a collective term for this vast cocktail of proteins and growth factors that these stem cells make. And the second part of this combination therapy is to combine this secretome with an injectable hydrogel. So hydrogels are 3D networks of polymer chains that hold a high water content. I like to think of them as fancy jelly. We think that using this hydrogel will act as a slow release drug delivery system for the secretome, allowing it to work for longer. And we also think that by using hydrogel, that will support the regrowth of damaged nerve cells in the injured spinal cord. I found that all the staff are really friendly and supportive. I feel like we have a really thriving early career researcher community. So we have a networking group called Envision, who organise a number of events which are principally designed to help our career development.
We are here at the QPCR core facility at the IMS. And here we have state-of-the-art technology where we use qPCR machine to analyze samples for gene expression. Now come with me. So, this is a qPCR core facility where we have several machines for the analysis of gene expression. As you can see here, this is a Roche light cycler machine 48-2. This machine has a 96 well block in it. What that means is this machine has the capacity to run 96 samples at one go, and it is connected to a desktop system where you could literally see the real-time amplification of your samples. Because of the sensitivity of this sort of technique, this particular section of the IMS laboratory is dedicated for the loading of samples. So as you can see here, we have our own special pipettes for this particular facility where researchers such as PhD students, postdoctoral researchers, or staff could readily bring in their samples for analysis. We also have specialized pipette tips which are sterile, because you need to have sterile pipette tips to run this sort of experiment basically. So aside that, we have also a well-standard centrifuge for the spinning of your samples as well. A qPCR plate looks just like this. You have the tiny bit of wells where the individuals is meant to fill in the samples. I know you may wonder, why should I touch this basically? This is just a balance well plate, whereby you could use this to balance the centrifuge while you spin your actual sample plate for analysis. But this is a typical example of what a qPCR plate looks like basically. So outside the Rush 4896 block machine, we have other varieties of machines as well within this facility. And again, we have the light cycler Rush machine, which is a 384 well block. So this simply means that this machine as well has the capacity to run 384 samples at one go. At the same time, like I explained before, this machine as well is connected to a computer where you can see the analysis of your samples in real time. Again, you have as well the Rush machine of 384 well plates blocked in. The same thing is applicable here, where you can run your sample at 384 well plates and at one go as well. This particular equipment as well is connected to the computer here for real-time result analysis. The qPCR core facility is so important in today's world of COVID-19. The reason for this is the fact that we all know that today we are plagued by COVID-19 pandemic. What that implies is, I'm sure you've heard over the news what is called qPCR analysis for COVID-19 test. Well, if you've been hearing about that all along, this is the sort of equipment we use to analyze such samples for COVID-19 virus out there at the moment. Again, because of our collaborative efforts of the university, specifically the Institute of Medical Sciences, we had to donate a few of our equipment to the NHS Grampian, whereby some of our staff, PhD students, undergraduate students as well, volunteered to support in mass testing of COVID-19 here at the NHS Grampian. The IMS is home to a number of research centers. One is the Aberdeen Cardiovascular and Diabetes Center, which is seeking to improve cardiovascular and diabetes patient care. Two is a Center for Arthritis and Musculoskeletal Health, who are developing new methods in prevention and treatment of arthritis and musculoskeletal condition. Third is the Aberdeen Biomedical Imaging Center, who are working to improve diagnostic imaging technologies. Fourth is the Center for Bacteria in Health and Diseases, who are trying to understand bacterial infection and the importance of bacteria in health. Sedaf Ashraf is a research fellow with the Center of Arthritis and Musculoskeletal Health. Dr. Sedaf will now tell us a bit more about the research they do.
My name is Sedaf and I'm a research scientist, and I work within the Arthritis and Regenerative Medicine Laboratory, which is situated within the Institute of Medical Science. And on the whole, we are a part of the Aberdeen Centre of Arthritis and Musculoskeletal Health. So I'm researching on stem cells, which are basically a group of specialized cells that build new tissues. And we are trying to look at whether these stem cells can be manipulated to control the progression of arthritis and whether we can induce joint repair. So I'm trying to understand the crosstalk between stem cells and nerves in an arthritic joint so we can alleviate pain associated with arthritis. Now, arthritis affects half of the world's population over the age of 65, and pain is the most debilitating symptom which affects people suffering from arthritis. So if we can understand this crosstalk, we can provide better treatments which can not only alleviate pain, but they can also improve the quality of life of the patients. And in order to do that, we are using cell culture, so we grow cells in the laboratory. And in addition to that, we're using patient samples to look at how we can translate the findings from a single cell model all the way into translation into patients.
COVID-19 has had a huge impact in our lives, and the research we do here at the Institute of Medical Sciences. And due to this, we've had to put in strict measures and regulations to ensure that researchers, staff, and postgraduate students are safe while in the building. However, the university has received grants to investigate treatment and response to COVID-19, and some of this grant is based here at the IMS. Professor Mirela Delibegovic will tell you more about our research on COVID-19.
My name is Mirela Delibegovic and I'm a professor in diabetic medicine here at the Institute of Medical Sciences. In May this year, we got the funding from the Scottish Government through the Chief Scientist's Office to allow us to develop a novel and reliable test to detect antibodies against COVID-19 in patient samples. So this is a collaboration between a spin-out company, Vertebrate Antibodies Limited, as well as NHS Grampian. And really the company's artificial intelligence software allows us to find regions on the virus that would be very specific against SARS-CoV-2 virus, and therefore allow us to develop this new test to know the prevalence of COVID-19 amongst the general population. One of the problems at the moment is that the current tests were never developed against SARS-CoV-2, but just coronaviruses. And what we are finding in our work is that these tests are not specific or reliable, because when we test some of the common cold samples, for example, they also come through positive. So we are really hoping that by the end of this year, we will have a functional test that is not just reliable, but also very specific against SARS-CoV-2. I think it's really important to know the prevalence of the infection in general population. So all of us will have had some kind of symptoms that we now associate with perhaps having had COVID-19, fever, cough, etc. And one of the problems is that the current tests, as I said earlier, are not very specific. So we may have some false positives and we may think we have already had it. It's actually quite important for general public to know the prevalence of the infection itself. So not only could we test NHS key workers and key workers such as teachers and care home workers, but also the general population, to not only know whether we have had it, but also if it gives us any protection going forward.
We hope you've enjoyed today's tour. The team at the IMS would have loved to have you on ground to tour the facility, but we believe today's online tour will give a view of some of the research we do here and the things that goes on within the building. Hopefully next year we will have you on campus to tour the entire facility, at the same time to update you some of the research we've talked about today. For now, thank you, and it's goodbye from me.